All right, so this lecture we're going to be talking about acquired immunity. Now, acquired immunity differs from innate immunity because innate, is immediate, it's right there, it's present all the time since birth, and it has no memory, it's non-specific. Acquired immunity is actually very, very specific. And the first star is that it develops over time. It develops days after exposure. So no, it is not immediately available. It is highly specific to an infectious antigen, unlike innate, which is non-specific, right? It improves upon repeated exposure. And the big star is that it has memory. But you need to remember that even though acquired immunity is so important, it does not and it cannot exist without a properly functioning innate immune system. So in order for you to get to step 2, which is acquired immunity, you must have a healthy step 1, which is innate immunity. Now this is mediated by antibodies, by B cells and T cells. And this is a very important concept. Natural killer cells, B cells and T cells, are all classified as lymphocytes. But remember that natural killer cells are innate immunity, or they fall under innate immunity. B cells and T cells, on the other hand, are cell-mediated or acquired immunity. But they're still all lymphocytes. And antibodies are, they fall under a sort of like a sub-branch known as humoral immunity. Now, what is passive immunity? It is any immunity gained from antibodies or lymphocytes of another person. And the most common example of passive is breastfeeding. All right, a mother passing on her antibodies onto her baby. Now, the advantage is that it's immediately available, but the disadvantage is that it has a shorter life, well, not even shorter, it has a short lifespan. No memory is made because for your immune system to have the memory, it must do the work. And the way I like to think about it is with passive immunity, think of 2 students taking a test. They're both taking an exam. You have Sammy and you have Tommy. Sammy studied all month for this exam. He is more than prepared. He has the memory. Everything is almost graved into his head. He can take the exam with his eyes closed. And then you have Tommy, who didn't really study at all for this exam. All right, and he plans on cheating and getting all the answers from Sammy and passing. Now, both of them may pass, but Sammy's the one who made the memory because Sammy's the one who did all the work. You have the same scenario here. You have the mother giving off her antibodies to her child. The mother has memory because the mother actually did work to make those antibodies, but the child is just getting them without doing any work. So the child is not going to have any memory. And a major disadvantage for passive is that they're at risk for hypersensitivity and anaphylaxis. Now, homologous refers to a pooled human immunoglobulin G for post-exposure prophylaxis. Now, in medicine, whenever we talk about prophylaxis, we mean prevention. And this is specifically after the exposure. So prevention after a person has been exposed to any of the following: Hep A, B, rabies, tetanus, measles, and varicella. And heterologous is an antitoxin that's used specifically for botulism and diphtheria, which we're going to get more into once we touch infectious disease. Now, the complication of heterologous is serum sickness and anaphylaxis. And then we have monoclonal antibodies. And you want to put a big star next to monoclonal antibodies because these are derived from a single B cell, and these are highlight this. They are specific for cancer. And when speaking of cancer, they're specific for specifically multiple myeloma, which is a type of blood cancer. But monoclonal antibodies are almost always associated with multiple myeloma, so you definitely want to keep that connection. And of course, they're also associated with autoimmune and infectious disease. Now we go into passive active immunity, and these are both preformed antibodies plus vaccines. So vaccines are your active, antibodies are your passive immunity, right? And this is given for tetanus, for rabies, hepatitis B. And active immunity is the production of one's own antibodies or lymphocytes against an antigen. And this results from 2 scenarios. Either you were naturally exposed to the antigen, or you were exposed to it through microbial products, also known as vaccines very commonly. Now, the major advantage of this is memory. All right, that's why vaccines are so efficient, because you make memory. And a slight disadvantage is that the primary exposure is going to take the longest time. by the time that second antigen comes around, or it comes around for a second time, your body's going to know off the bat that they need to kill this because they already have the memory. Live attenuated are produced by modifying a wild microbe, and they retain the ability to replicate and produce immunity, but they usually do not cause illness. Now, of course, in an immunocompromised person, they most certainly can, but in an immunocompetent person who is otherwise healthy, they usually do not cause illness. They just replicate and produce immunity. All right, and the immune response is very similar to the natural infection, and this is usually effective within a single dose. And the most effective immune responses are produced by the live antigen. So examples of the live attenuated, you have the MMR, varicella, zoster, yellow fever, rotavirus, intranasal influenza for viral, and you have BCG and oral typhoid for bacterial. Now, inactivated are whole or fractionated viruses or bacteria that are protein or polysaccharide based, and the star here is that they cannot replicate or cause disease even in immunodeficiency. So if you have an HIV patient on the floor, this is the vaccine you want to give them. You want to give them this inactivated vaccine because even though it's a fractionated virus or bacteria, it just cannot replicate. And you know that if a microbe cannot replicate within you, it most certainly cannot cause disease, right? And this is usually effective within 3 to 5 doses. And the immune response is using antibodies, hence humoral. Examples of inactivated include polio, hep A, B, rabies, influenza, acellular pertussis, and HPV for viral, and diphtheria and tetanus for bacterial. Now, polysaccharide-based are T-cell independent, and they are not immunogenic in children less than 2 years because the children aren't mature enough. Their immune systems aren't developed properly. And it's because what we call of immune immaturity. There's no booster response with repeated dose, and there's a less functional antibody, usually only immunoglobulin M. Examples of polysaccharide-based vaccines include Salmonella typhi. And for polysaccharide and conjugate vaccines, you have pneumococcal, meningococcal vaccines.